Hello, my name is Łukasz Rączkowski and I'd like to present this poster titled Deep Learning-Based Tumor Microenvironment Segmentation is Predictive of Tumor Mutations and Patient Survival in Lung Cancer. In clinical practice, hematoxylin and eosin-stained tissue slides are commonly gathered for patients during biopsies. As a result, there is an abundance of image data that can be used to study the tumor microenvironment. The structure of that microenvironment is of crucial importance for patient diagnosis and survival. That's why in recent times, there has been a lot of work on different predictive models that try to find relationships between the tumor microenvironment encoded in tissue slides and phenotypic outcomes. However, most of these models work as black boxes, and as such, they're very hard to interpret in terms of biological features. Consequently, there is a need for models that use bespoke human interpretable features, which can be used to gain new insights into the tumor microenvironment. In this work, we introduced new human interpretable features calculated from segmented lung cancer tissue slides and used them as predictor variables in the tasks of survival prediction and mutation classification. The first step of this work was the training of our deep learning tissue classifier called ARA-CNN within an active learning feedback loop. We were provided tissue slides for 57 lung cancer patients, which were annotated by an expert pathologist. Then we split the annotations into small tissue patches and put them together in a training dataset, which was used to train the model. After that, we measured the performance of each of our 9 classes and asked the pathologist to annotate new regions for the worst performing ones. We then extracted new patches from these regions, added them to our training dataset, and retrained the model. We repeated this process 3 times, which gave us very good overall classification accuracy. The dataset generated in this process will be published, so the community will be able to train their own classifiers. As a next step, we downloaded lung cancer tissue slides from the TCGA database and processed them in order to extract human interpretable features. Each slide was split into tissue patches, and then each patch served as an input to our trained ARA-CNN model. This gave us classification for each patch, which upon combining resulted in segmented slides. Then we calculated our special features from these segmentations. The first of these features is TIP, which stands for tissue prevalence. It's a vector of proportions where each component is the number of patches of a given tissue divided by the number of all patches in the slide. The second of our features is TMEC, which stands for tumor microenvironment composition. Similarly to TIP, it's also a vector of proportions, but this time each component is the number of patches of a given tissue located specifically in the tumor microenvironment, divided by the number of all non-tumor patches in the tumor microenvironment. The TME is defined as a margin around areas where tumor tissue neighbors other tissues. With these features calculated for each lung cancer slide from TCGA, we also downloaded clinical and mutation data for the corresponding patients and used them together in the task of survival prediction with the Cox proportional hazards model. The models that used our TIP and TMEC features both resulted in C-index of over 0.7. We plotted hazard ratios for these models and observed a few interesting characteristics. In general, we can see that the presence of immune cells and mixed stroma tissue in the whole slide improves patient survival. The same is also true for the presence of immune cells in the tumor microenvironment, but to a lesser extent. When it comes to features that negatively impact survival, these are necrosis in the whole slide and both necrosis and vessel in TME. What's important, these last results are statistically significant. The second task was the task of mutation classification. We selected a list of genes that are known to be relevant in lung cancer, and for each of them trained a binary classifier in order to predict if it gets mutated. For the training data, we used clinical data from TCGA and our TIP or TMEC features calculated from TCGA slides. We got AUC over or close to 0.7 for two genes, PDGFRB and RET. For these genes, we performed future importance analysis in order to get an insight into which tissue features are the most impactful. For PDGFRB, The most impactful of our features are mixed stroma in TMEC, vessel and immune in TIP, followed by bronchi in TMEC. For these 4 features, we plotted a value distribution for patients with mutated and non-mutated genes. Out of these 4, only vessel in TIP is statistically significant. As for RET, the most important features are bronchi, mixed vessel and stroma in TMEC. Moreover, we can see that all of our TMEC features are more important than clinical features. In terms of the value distribution, mixed and bronchi are statistically significant. To summarize, we trained our ARA-CNN model for tissue classification in lung adenocarcinoma. In addition, we also generated a new training data set for tissue classifiers, which will be shared with the community. With our trained model, we segmented lung cancer slides from TCGA and computed our human interpretable spatial features, TIP and TMEC. These were then used in the tasks of survival prediction and mutation classification, and in both cases performed well, giving SC index of up to 0.718 in survival prediction and AUC of 0.735 for classifying mutations in the PDGFRB gene. Finally, our TIP and TMEC features are easily interpretable and as such give interesting insights into the tumor microenvironment. Last but not least, I'd like to thank my supervisor Eva Szczurek for all of her support and all of our collaborators for cooperating on this project. Thank you.